Welcome to the program of Hands-on fNIRS for Motor and Auditory Research. My name is Mariana Ceci and I will be your host for this lecture. This program was brought to you by the Santos Dumont Institute, the Federal University of Rio Grande do Norte, the University of São Paulo, the State University of Campinas, the Federal University of ABC, NIRx Medical Technologies, and Brain Support. Today, we'll enjoy the lecture of Silvia Benavides Varela, who is an assistant professor at the University of Padova in Italy. Mrs. Varela teaches at the Department of Developmental and Socialization Psychology. She teaches courses on fNIRS techniques, cognitive development, and developmental psychology. Her research lines are cognitive basis of human development and education, language learning and processing, and bilingualism. Let us all welcome Mrs. Benavides Varela.
Speakers. I'm very glad to be here, and I will probably skip also some parts of the presentation in order to actually finish on time. So sorry for this, and of course I'll be happy to answer any questions if there are any. I was just trying at some point to mention the fact that having a great advance in cognitive neuroscience contaminated positively also the progress in developmental cognitive neuroscience. That allowed us to basically start having some reflections about some of the mechanisms that allow the baby also to carry out some specific functions, to carry out some specific tasks. But what I was trying to say before is that not all imaging tools are equally well suited for infants and children for a number of different reasons, including safety concerns and the fact that it is very difficult to keep a baby without moving or motionless for extended periods of time. Sometimes you have to have some compromises, and sometimes you have to choose the technique in order to basically answer to the hypotheses or questions that you had from the very beginning. And this, of course, you have to take into account not only the theoretical part, but also the practical aspects. So you have to consider the population under study. And this is important for us that now we are talking about babies and auditory studies. And of course, the availability of financial and human resources that are necessary to effectively use it. I was saying that PET is one of the techniques, neuroimaging techniques, that usually is not used in cognitive neuroscience for basic research because of the safety issues that raises the fact of injecting radioisotopes in the bloodstream of the baby. So it is considered invasive and only used for clinical purposes. fMRI instead is a technique that is used and that provides really fascinating and highly informative work in infant research. However, this work is limited, is restricted to the study of sleeping or sedated or very young infants and to resting state analysis. Whereas NIRS, on the other hand, allows participants to be awake, alert, and interacting with the task or the stimuli. And this is, of course, particularly useful if one wants to understand the youngest developmental populations and how their brain systems are involved in particular online tasks and stay constant or change with maturation, experience, and learning. So compared to NIRS, fMRI has some disadvantages, beginning with the fact that, as I said, babies have to be motionless, but also the fact that NIRS, as opposed to fMRI, is completely silent. So NIRS is advantageous in that sense, providing non-intrusive environments and allowing easy presentation of auditory stimuli. Of course, another potential issue is the fact that some families are concerned with the fact of having their baby within the magnetic field, and so this decreases their the amount of volunteers that actually accept to participate in the studies. In terms of the signal, NIRS has the advantage of measuring both oxy and deoxy, while the pole response in fMRI is related to deoxy only. And so in some circumstances, the possibility to detect these two chromophores allow for a more detailed exploration of the precise metabolic hemodynamic processes. NIRS is also cheaper compared to fMRI, and this, of course, allows many more researchers doing basic science who do not have usually access to big fundings that are required for other imaging studies. And of course, we need to be aware of the fact that the key limitation of NIRS is that it only probes the surface layers of the cortex. And so the brain structures that lie deeper or below the cortex are not visible by most NIRS techniques. And of course, the spatial resolution of NIRS is inferior to that of the fMRI. So until now, I hope that you could have heard what I said. This is what I stopped before. So recent technological advances in NIRS allow to test participants, as I said, not only because they don't have to be rigid, stabilized in their head and the body, but also they can actually interact. And this turns it even more interesting for people that study social interactions in babies, for example. And so more ecological conditions such as free viewing and free flowing interaction can actually be also studied using this technique. So altogether, these features provide more flexibility to choosing the experimental setting compared to other techniques, and of course guarantee more comfortability to the participants. So much that they can be used with the youngest preterm and premature infants. In fact, NIRS is one of the techniques of choice in studying brain functions in these vulnerable groups whose skin is very thin, physiological functions are immature, sometimes weak, and sometimes even pathological. And yet, NIRS is a technique that they can tolerate for even long-term monitoring of brain oxygenation and function. Another huge advantage of NIRS is its portability. It's a very compact device, so it means that it can be transported or used and set in different laboratories or research settings. And this extends the applicability of this method to a range of scenarios and uses, including bedside medical monitoring and research outside the traditional laboratory settings. For example, NIRS can be taken to the homes of the participating infants for studying their behaviors in familiar environments, and reaching even geographical areas where the conditions to carry out a study might be otherwise very difficult, as has been nicely demonstrated by the work of Sarah Lloyd-Fox, for example. For many years, before NIRS became popular, the primary choice for functional imaging in awake infants, and particularly to carry out auditory studies, was EEG. So another non-invasive techniques that was actually introduced before, and that probably most of you already know, with high temporal resolution but relatively poor spatial resolution compared to NIRS. And of course, the arrival of NIRS, which provides a better spatial localization, was a welcome addition to the initial limited choice of neuroimaging methods that were suitable for the use of infants. In the talk that follows mine, you will actually have a better idea of some of the applications that have been recently developed to combine both EEG and NIRS, particularly in the experience of Judith Gerben, a colleague here in Padova. So the question one can ask is, what has been studied with electrophysiological measures, and what additional information can one obtain with higher spatial resolution provided by NIRS? As probably some of you already know, this long tradition in EEG studies has brought significant contributions to study many aspects of auditory sensation and perception, including sound localization, sound discrimination with the central auditory pathway, but also more sophisticated processing in language acquisition studied with infants, for example, by identifying components associated to semantics and grammar at different ages. However, the assumptions inherent in the solution of the inverse problem for source localization continue to restrict the ability of this technique to precisely characterize other equally important organizational principles of auditory processing. And I'm talking here about the combination of features that must be present for a given complex stimulus to stop being perceived as merely a sound and instead began to elicit some responses from different regions that are responsible for the detection of categories, say species-specific vocalizations like speech in humans. Many studies have demonstrated in that sense that NIRs can be properly used to study how language, music, and other sounds are represented, and how these representations change from a developmental perspective across different age groups in both first and second languages, and also probing theoretical questions more deeply, obtaining not only localization of functions, but also more mechanistic explanations of the phenomenon. And so by providing better spatial resolution, NIRs, in this case, studies in adults have revealed, for example, that different types of sounds may show different cortical activation patterns. And so what I'm showing here are the results presented by the study of Hon and Santosa. And the authors observed varying patterns of activation in the left and right auditory cortex in response to speech sounds, nature sounds, annoying sounds, etc., which could be identified and teased apart also by classification algorithms. And importantly, this type of analysis, even with finer spatial resolution, can be performed using also fMRI. However, the mechanical noise produced in the MRI is problematic and reduces the classification accuracies compared to those reported in NIR studies. We know that researchers have and are exploring ways to minimize the impact of MRI scanner sound in auditory research, but to date, the silent devices of NIRs continue to be much more simpler to use for these purposes. So sound discrimination, classification in general is one of the advantages that this technique offers as compared to other techniques in the auditory research. Another study of sound discrimination was nicely carried out by Poloni and collaborators, who showed that fNIR signals provides an objective measure of discrimination. It discriminates whether an individual is hearing normal or distorted speech. So what they did is to test activation within the auditory cortex in 19 normal hearing subjects while they listened to speech with different levels of intelligibility. Normal speech reliably evoked the strongest responses within the auditory cortex, as you can see. Distorted speech produced less region-specific cortical activation, and environmental sounds were used as a control, and they produced the least cortical activation. And so this also shows that fNIRS data can, as I said, discriminate between the brain's processing of good or poor speech, or one can say also clear or unclear sounds. That brings me to another advantage of using NIRs in auditory studies. Indeed, until recently, measurement of cortical activity after cochlear implantation was restricted due to the incompatibility of most neuroimaging methods with cochlear implants. For example, many types of implant must be surgically removed before MRI scanning, and the implant electrical components are not safe to enter into the magnetic field. So this not only poses additional risks for the person, but also means that the participant cannot listen to auditory stimuli during scanning. And so some MRI-compatible implants are now available, but they can also interfere or distort the signal, particularly the one coming from the temporal areas where the auditory cortex is situated. So that's a problem. And also it's a problem for measurements of in EEG or MEG recordings, where electrical and magnetic artifacts can contaminate the data. And this is not a problem in NIRs. And so NIRs is compatible with this and other hearing devices because it is not sensitive or particularly sensitive to electrical signals. And so this is a technique that is suitable for clinical purposes, of course, but also for research purposes. For example, when exploring cortical plasticity following recovery of this important sense of hearing, and for comparing cases of, say, congenital deafness and individuals who develop deafness later in life, for example. So now let me explore a few examples. Of course, this is not review, but just a few examples of studies that have used NIRs to understand how infants get to efficiently process the sounds that form language. And I will focus specifically on some processing abilities observed in newborns and which reflect the initial predispositions to attend and discriminate language from other sounds, which somehow resonates with the type of studies that I was mentioning before in adults. But to talk about babies, let's consider the experience of the baby, the human newborn, and imagine their very first reaction when listening to another human being speaking to them, now that they are finally outside the womb. So the question is, are babies somehow ready to process speech as if it was a special signal for them? Is their brain capable of discriminating language from other sounds, even if they have not had enough experience with this sound? Answering this question has theoretical implications, as it somehow contributes to these long-standing debates between nativist and constructivist approaches to language development. So we can basically ask here, are babies in their first 1, 2, or 3 days of life equipped with a device capable of learning language from birth, or do they require a significant experience in order to develop their linguistic abilities? And so by using NIRs, studies have been able to explore precisely these kind of questions. Can babies react differently to the sound of speech and to other sounds? And this was the aim of the pioneer study of Marcela Peña and colleagues, one of the first studies in auditory processing using NIRs in infants. In their study, the authors presented newborn babies with, presented here in A, so normal infant-directed speech that was played for voice. That is, short sentences uttered by a person at a typical loudness and speed. This is normal speech. Of course, the sentences, as it is common in NIRS designs, were separated by silent pauses of varying durations in order to avoid any response that would be locked to a given frequency that could be filtered later on, and that could be contaminated with other sort of frequencies and other sort of signals that need to be filtered later on. So the sentences had these silent pauses in between, and brain responses to these conditions were compared to those elicited by backward speech. So that is the same precise utterance that play in reverse. So let's note here that in this condition, the physical auditory stimulation remains the same, but the transitions of the sounds are different, and speech becomes unintelligible even to adults. So, and so they had also a third condition, which is the condition here in C, which is corresponds to silent or no auditory stimulation. And here to the right, what you see are the results that the authors reported. So you have the groups or the optoid positioning, which is, of course, over the auditory area in the temporal left and right hemisphere. And what you see here are the channels, and basically the representations in red would correspond to what they reported is total hemoglobin concentration. There was also a report on oxyhemoglobin and deoxyhemoglobin. Basically, the authors found significantly more activation when infants were exposed to normal speech than to backward speech or silence. And so the normal speech here represented in red, and the other two conditions in green and blue. So this suggests that indeed speech is treated as a special signal. And in addition, the responses were observed in the left temporal area, as you can see. And so because these areas are homologous to those that mediate language processing in the adult brain, the authors concluded that neonates are born with this lateralized or left hemisphere superiority to process specific properties of speech, and that they might not require a long life experience with language to show this lateralized response that is typically observed in adults. Subsequent studies have revealed that newborns not only detect speech, but their brain also discriminates different configurations within speech that might be relevant for understanding the processes that guide language acquisition from the beginning. So it is well known, for example, that across languages, certain syllable structures are more common to others. For example, syllables like blif are preferred to syllables like diff and bif. But whether such regularities reflect, say, historical processes or production pressures, because they are more difficult to pronounce, as you probably heard. So diff and bif are more difficult to pronounce than blif. So maybe this is the reason why they are not preferred across languages, or maybe this reflect a universal linguistic principle. And this is the kind of question that one can try to answer by looking at babies that do not have experience and that, of course, do not pronounce yet any syllable. So in this study, David Gomez examined whether some precursors of these preferences are already present early in life. And as you can see, the baby's brain responds differently to syllables that were well-formed or ill-formed. So you see the two type of brain responses to the well-formed and ill-formed syllables in two experiments, actually. So that was replicated using different configurations, but just basically showing in the left inferior region of the brain that the baby is actually capable of reacting differently to configurations within speech. And this suggests that there is possibly an early universal bias that helps shaping language acquisition. Newborns also seem to possess abilities to detect basic structural irregularities in speech that imply analyzing the relations between different elements of an utterance. So not only detecting the structure within a syllable, but also the relations that the different items within sentence A will have. For example, the study of Judith Jerven and colleagues presented newborns with utterances which had simple regular patterns. In this case, as you can see, a regular pattern would be the repetition of one of the elements at the end. So one element that is different from the second one, and the second and the third are the same. And they compared this to stimuli in which there was no repetition, so there was no structure that the baby could eventually pick up. And so what you see up here in the graph is the representation in, say, pinkish, the activation response to the simple structure in oxyhemoglobin concentrations, and to no structure in red here. And you see that the authors found greater activation in the temporal and left frontal areas, which are marked here. So these are the temporal in the left and right, and here the frontal areas of the left hemisphere to sentences implementing structures than to sentences with no structures. So babies can keep track of regularities in the speech. And this is reflected in the brain responses. And this is what NIRs is telling us here. In addition, newborn brains encode basic information that might be necessary to keep track of the relative order of words within a sentence. And this is very relevant because the order of words in a sentence carries important grammatical functions. So in this study, we basically compare the responses of a baby to sentences that had a word order that did not change, so in the case of red here, and word order that did change, in pinkish. And you see that newborns show differential responses to these sentences, so those that maintain the same word order compared to sentences with variations in word order in areas of the brain that have been associated with language and syntax processes in adults. So altogether, these findings suggest, in a sense, that newborns are sensitive to certain input configurations when processing speech signals, and then these abilities might facilitate later language development. So we might say, in a sense, that indeed the human brain is an optimal language acquisition device since birth, and that is predisposed to attend to certain input configurations of speech. But one can ask, and some of you may be even wondering whether these abilities reflect a biological feature of human species or whether they derive from the experience that babies might have received, for example, in the womb. Before looking at some studies that have explored these questions, it is important to keep in mind that hearing begins in the second semester of pregnancy. However, the perception of the sounds is different, not only due to the immaturity of the auditory nervous system, but it is also limited by the mother's physiological noises like heartbeat, respiration, digestion, that make the environment of the womb quite noisy. And in addition, the maternal tissue and amniotic fluid act as a filter for the sounds and allow only low-frequency components to be transmitted to the feature, so rhythm and prosody mostly. Even if filtered, it seems that the information that arrives to the fetuses is sufficient to explain some responses of the babies at birth. For example, newborns show strong preference to listen to their mother's voice and to the rhythm of their native language, and their brains respond differently to native versus non-native language, suggesting once more that the experience with the native language gained in utero influences how the newborn brain responds at birth. But what happens when the fetuses do not receive this kind of filter information in the last weeks of pregnancy, but instead they receive a broadcast speech outside the womb? Lisa Bartha-Dorin and their colleagues in Austria recently investigated this question using a paradigm similar to the one proposed by the original fNIRS study on language discrimination by Peña and colleagues, but this time with newborns born preterm. And the results indicated that, as opposed to healthy newborns babies who show clear discrimination patterns between forward and backward speech, preterm babies do not show signs of discriminating between speech and non-speech at term equivalent age, that is, when reaching levels of maturity similar to those of the full terms. Moreover, gestational age at birth positively correlates with the discrimination abilities in preterm babies, such that the later the baby was born, the better the discrimination abilities. So it seems that experience with language outside the womb does not explain language discrimination capacities at birth. Otherwise, preterm infants who have many more weeks of experience with language outside the womb would have performed better than full-term neonates that have been just exposed to broadcast speech. Instead, as you can see here, the opposite was observed. So an interesting possibility is that the filtered speech sounds that mostly contain prosody and that reach the fetuses in the last weeks of pregnancy played a crucial role in shaping the first stages of language processing in typical language acquisition. But this is, of course, a question that we explore further. Different from this filter information that gets to the baby in the womb, we have some distinctions that are imperceptible there. And so it is only after birth that the newborn brain has access for the first time to this acoustic detail that distinguishes all the words in the surrounding language. An important part of my research has focused on unveiling this initial state of memory capacities in humans and the properties of the memory networks that support the encoding and recognition of the very first word memories, how detailed they are, and which brain areas are implicated in these mechanisms. So I will continue speaking. It's already past 3 now here. So I don't know if I have time to go ahead. It's been 30 minutes that I speak. So I don't know if can someone from the organization tell me if I can go ahead or if you want me to stop.
You can go ahead, Silvia, please.
Okay. Thank you. Thank you so much. So our memory studies, a typical NIRs design in our studies will include a familiarization phase of 6 minutes, a retention interval of 2 minutes, and a test phase of, say, about 3 minutes. Familiarization and test phase are organized, as you can see, in block design or block fashion. And during the familiarization, all the babies are exposed to the same word, whereas in the test, half of the newborns hear the same word they hear during the familiarization, and the other half will hear a word that is completely novel. This is typical sort of recognition response that we will observe in this kind of studies, very similar to those that one would have expected even with other type of techniques. So here on the x-axis, you see the number of blocks, and in the y-axis, just the differences. So oxyhemoglobin, in this case, concentration changes. And so what we observe here is this differential response in the first block of the test phase, such that there is an increment in the hemodynamic response when babies hear a novel word, and a decrement in the hemodynamic response when babies hear the same word they hear during the familiarization. And this signature of recognition is usually found in the right frontal area. This area is usually activated in adults when retrieving verbal information from memory or during recognition. And it is also activated in 3-month-old infants when they repeatedly listen to the same sentences. So the activation of these in newborns was, in a sense, taken as a confirmation or double evidence for the presence of mechanisms of recognition supporting the first stages of language acquisition. However, newborns' memories are not particularly detailed. So we knew from behavioral studies that newborns are capable of perceiving all the phonemic contrast of the languages of the world. But in a NIRS study, we observed that they memorize only some segments of the words. In particular, they start with a bias towards encoding the sound of vowels and not consonants. And this is what we can observe in this graph. As you can see, there was again an increment in the mean hemodynamic response that once again signaled novelty in our paradigm when babies heard in the test a word that had different vowels compared to that in the familiarization, but preserved its consonants. So it's as if the consonantal information preserved was not sufficient for them to recognize the sound as familiar. Congruently, when the vowels were the same, this is what you see here, vowels are the same, but the consonants change. The newborn's brain show a pattern compatible with the recognition of the familiar word, once again showing that segments that carry most of the energy and transmit most of the prosodical information in speech, namely vowels, are better encoded and recognized by infants. And this was, of course, found once again in the same right frontal area, and this is how our hemodynamic response looked like. By applying other analyses, we were able to further describe some of the patterns across time and interactions among brain areas supporting memory representations at birth. So what you can see in the upper graph here are mean concentration changes once again over time, and this time we are focusing on the left frontal region. We have here mean responses in the y-axis and the 10 familiarization blocks of the encoding phase in this x-axis. And regression analysis confirm an adaptation or habituation-like hemodynamic response, which is clearly visible as the mean intensity gradually decreases over time. What is interesting here is that this decrement in the local hemodynamic response is concomitant with the decremental strength of the effective connection between this area, so once again, the left frontal area, and the left temporal area. So here you have that basically encoding new information, which starts in the first block, implicate an interplay between frontal and temporal regions in the newborn's brain, and this interaction diminishes. You see the path coefficients diminish as the information becomes less and less novel, or which is the same, more and more familiar to the baby. If this is the case, one will expect similar strong interactions between these two areas in the test phase when babies hear a novel word, and this is precisely what we observe. So once again, as you see, only the group of babies that hear a novel word show these strong interactions between frontal and temporal areas. On the other hand, notice that during recognition, so we had, as I said before, the right frontal area is the area that generally shows the differential hemodynamic responses. So the strongest effective connections of the right frontal area here are with the right temporal areas only during the presentation of familiar sounds. You see, this is observed during the presentation of familiar sounds, but it's not observed during the presentation of novel sounds. So what we conclude here is that there is a coordinated activity in the neocortex that might be characterized by the interplay between temporal and left frontal regions during encoding and between temporal and right frontal regions during recognition of words. Besides describing these, I think, interesting interactions in the neocortex, we also found that even though memory representations at birth are not detailed, also talking about the fact that they focus mostly on vowels, they can be rather specific to the properties of speech. Indeed, the newborn brain, while capable of, say, retaining a memory trace during the silent retention interval, or even after listening to music, they actually tend to forget the trace when new words are presented in the interval between the learning and the test phase. And so this suggests, in a sense, that a newborn's verbal memory might be stored in a representational code that is selective enough to receive interference from other speech sounds, but not from music, and that verbal memories can be fragile in some circumstances. Which are those circumstances? At the moment, we are trying to understand how these early memories can be stored for longer periods or preserved in a more endurable ways, and how this is reflected in the reorganization of the neural circuitries. I might take the opportunity here to announce that I will be opening some postdoc positions in the lab associated to a recent grant from the European Research Council that I obtained, and that I will be precisely focusing on studying these questions related to memory capacities in babies. So if any one of you is interested, please just drop me an email and I'll be happy to talk about this. So I hope I have convinced you so far that NIRS is a reliable method to study auditory functions in adults and children. And as a neuroimaging technique, it's that is generally suitable to study very young infants. What I have not yet told you, but probably some of you already know, is that brain imaging with babies and infant studies in general imply important challenges from an experimental viewpoint, and which are important to acknowledge and possibly also taken into account when designing an experiment. So first, if you are testing a baby, the first thing you should know is that their attention span is quite limited. That is, they will look or pay attention to the visual or auditory stimuli wherever you present and remain still, but only for a few minutes. So on average, I will say 5 to 7 minutes maximum on average. There might be some exceptions, of course. And in general, this implies fewer trials compared to adults, lower signal to noise ratio. And of course, this limits a lot the kind of analysis which cannot be too sophisticated due to, again, the lower amount of data. Of course, this gets even worse if one has a conservative approach to data analysis and decides to reject all the trials that contain any emotional artifacts instead of attempting any correction. The exceptions to this problem or this problematic data in infants are usually newborns, luckily, namely babies of 1 to 3 days of age, who may also sleep or stand longer recording sessions of up to even 20 or 30 minutes without moving a lot. One possible recommendation to avoid losing too much data, or in case you need longer recording periods for your study, is that you might plan your experiment in such a way that there can be ways out or pauses as needed. So moments in which you can entertain or make the baby feel relaxed and avoid her or him getting fussy too quickly. There are also some methods available to avoid rejecting data, so avoid rejecting trials with motion artifacts by applying, for example, wavelets as some of the recent recommendations of even Sabrina Brigadoy here in Padova are actually suggesting in her studies. In auditory studies with older infants, it is also common to play silent movies or cartoons for the babies while they listen to the sounds that are being tested. And this is, again, with the main aim of maintaining their attention. Of course, it is very important to carefully choose this stimuli to avoid any possible confounding with your data. Another situation that might be very frustrating, but you have to know it, is that babies can cry in the middle of the study or simply stop collaborating without completing a minimum number of trials that you have actually decided for your study. And this means a lot of working time without being able to effectively collect data. In NIRS studies with infants, the attrition rate can vary a lot from one laboratory to the other. It could range from about 25 to up to 50 or 60%. And of course, it can depend on a number of factors, such as the age of the participant, the properties of the stimuli, the expertise of the experimenters, and so on and so forth. So it is important that when you calculate the sample size for your study and you plan for a chronogram or for your recruitment, you take into account these important attrition rates and keep a record of them for future calculations. Like in any other baby experiments, it is also important that parents who are usually holding the baby are not aware of the specific research question, or at least to make the stimuli unavailable to them by, for example, playing music through headphones to mask the actual sounds the baby is hearing, or make them wear, say, dark glasses when testing visual stimuli. Another condition or situation that you should be aware of, and I will close here, is the variability of the hemodynamic response function, which is huge in babies. We know how the HRF should look like, at least in theory. We are supposed to observe increments in HBO and decrements in HB, but in actual practices, we might find deviations from the canonical patterns, and even the opposite patterns might appear. And these deviations can be very hard to interpret from a biological standpoint, and even more for interpreting theoretical implications of the results and for driving conclusions. So this is something that we should keep in mind. And the last point that I would like to highlight today is that there might be some dishomogeneous developmental patterns of the HRF, that can be observed across various areas. But one can generally expect that within the auditory areas, which are the focus of this presentation, the mean latency of the maximum peak of the activation will decrease in the first months of life. So I'm talking about the latency of the maximum peak that will decrease with age here, whereas the amplitude tends to increase. So as you can see, it tends to be higher in the graph. And these variations, of course, should be taken into consideration, for example, when analyzing or interpreting data from various age groups. So I will finish my presentation here, and thank you very much for your attention. Apologies once again for the difficulties we experienced before. Thank you.
Thank you very much for your time. We hope this lecture was useful to you, and please don't forget to subscribe to the Santos Dumont Institute's YouTube channel so that you can have access to these and many other contents related to neuroscience, neuroengineering, and research. Thank you.